Hello everyone, my name is Konstantinos Stegopoulos and I am a PhD student at the Department of Molecular Biology and Genetics in the Laboratory of Molecular Developmental Biology and Molecular Neurobiology in Alexandroupolis, Greece. I would like to welcome you to Microbiome Applications for Functional and Sustainable Food Systems. We will discuss the latest trends and challenges shaping the dynamic landscape of the food microbiome by exploring and exploiting microbiomes in food systems. To begin with, let's define what microbiome is. The term microbiome refers to the diverse community of microorganisms, including bacteria, viruses, fungi, and archaea, that inhabit a variety of environments, like the human body, animals, plants, and the surrounding ecosystems. Actually, they are everywhere, even in our foods. To give an example of their significant role, microbiomes support soil health, crop productivity, and protect livestock from diseases and food poisoning. We can say that microbiomes affect the overall health of entire ecosystems, and they possess key roles in the concept of connecting human, animal, and environmental health, which is also known as the concept of the One Health. Today, we will focus on the highly diverse food microbial communities, which comprise of countless bacteria, fungi, viruses, and archaea, with all of them forming distinctive patterns across different foods and contributing to the development of their unique characteristics. This is the main reason why, over the past 15 years, research on food microbiome has rapidly advanced, shedding light on the complex microbial communities that reside in our food. We have now uncovered the diverse composition of these microbial communities and have a deeper understanding of how they impact food safety and fermentation processes. This knowledge has led to several innovations, including food preservation methods, the development of functional foods, and the creation of microbiome-based technologies to enhance food quality and safety, promising a healthier and more sustainable food future. Probably the most significant trend that has allowed us to gain a better insight on both the composition and roles of these microbial communities is the continuous advancement in sequencing technologies, and especially the high throughput methods. Moreover, new methods to study microbiome, the so-called omic approaches, have drastically changed the way we describe and analyze food microbial communities, and now allow scientists to characterize microbiomes with great depth and resolution. The first omic technique is metagenomics. Metagenomics is a method that enables scientists to analyze the collective genetic material present in a sample. The advancement of high-throughput methods has made possible to sequence the entire microbial genome within a sample, and researchers can now uncover the identities of bacteria, fungi, and viruses that inhabit different food ecosystems. And thus, metagenomics provides information for the microbial diversity within foods. On the other hand, metatranscriptomics focuses on the RNA transcripts produced by microbial communities, allowing not only to identify the genes that are present in food-associated microbes, but also to assess their activity levels and functional roles. This way, we can better understand the dynamic behavior of food microbiomes over time, as we get a wealth of information about microbial metabolic pathways, enzymatic activities, and responses to environmental stimuli. Similarly, another omic technology called metaproteomics focuses on the study of the proteins produced by microbial species and offer a direct window into the metabolic functions and physiological activities of the studied microbes. Focusing on the proteins allows scientists to identify microbial enzymes, which are involved in a variety of food-related processes, like fermentation, degradation of organic matter, and synthesis of bioactive compounds. Last but not least, metabolomics. Metabolomics offers information for the metabolites produced by foodborne microorganisms, and thus provides valuable insights into their metabolic activities and interactions within the food ecosystem. By analyzing metabolic profiles, of microbial communities, we can identify biomarkers of food quality, safety, and spoilage, and bioactive compounds that may promote human health. All of these methods are rapidly evolving and provide scientists with a wealth of information, which can be used to facilitate the optimization of food production processes and the development of novel, functional foods with health-promoting properties. By combining data from different omic analyses, we can unravel the complex web of molecular interactions that govern the structure, function, and dynamics of food microbiomes. This, let's say, multi-omics approach provides a powerful framework for predicting and manipulating microbial behaviors, and it can help us optimize food production processes and develop innovative strategies for food preservation, safety, and of course, quality assurance. Using omics methods to describe microbial communities represents a major trend, and it is a transformative approach in food microbiome research, as it enables scientists to unlock the full potential of microbial communities within the food ecosystems and harness the beneficial properties of these microbes to improve human health, food sustainability, and global food security. The enhanced understanding of the complex interplay between the food microbiome and human health has given rise to microbiome engineering, a trend that involves the development of strategies for the optimization of the structure and hence the function of microbial communities within different food matrices. The innovative field of microbiome engineering encompasses various interventions that aim to modulate the food microbiome, to promote health and enhance nutritional outcomes. One such intervention that you may have heard about is the integration of beneficial and live microbes into food products. We call these microbes probiotics, and several strains of Lactobacillus or Bifidobacterium have been described to possess probiotic activity, and thus they can be incorporated into fermented foods like yogurt, kefir, or into functional beverages, even dietary supplements, in order to be delivered to the gut microbiota, so as to contribute to the microbial diversity and promotion of gut health. But apart from probiotics, microbiome engineering also encompasses the use of prebiotics, which are indigestible dietary fibers that serve as a substrate for beneficial microorganisms and selectively stimulate their growth and activity. Similar to probiotics, foods that are rich in prebiotic fibers can also be incorporated into the diet to promote the proliferation of beneficial microbes and improve the intestinal barrier function. This rapidly accumulating knowledge can help us start designing synthetic microbial communities. These communities can be used to develop novel food products with desirable characteristics, or they can be used for target modulation of specific microbial taxa or metabolic pathways within food matrices to optimize the nutritional profile and the functional properties of foods. This targeted modulation that involves the selective enrichment or depletion of specific microbial populations can help scientists to engineer foods with tailored microbiome structure and enhanced health-promoting properties. These health-promoting properties have been mostly associated with one of the most significant microbial communities within the human body, the gut microbiota, which comprises trillions of microorganisms and plays a vital role in modulating host physiology, metabolism, and immune function. We know that changes in the structure and function of the gut microbiota have been associated with a wide range of health conditions, like metabolic disorders, autoimmune diseases, or even neurological conditions. These findings have led scientists to use multi-omics approaches to unravel the complex interplay between gut microbiome, food, and human health. Overall, we can say that probiotics, prebiotics, and synthetic communities can be used for therapeutic interventions that are focused on modulating the gut microbiota and aim to develop personalized treatment strategies to individual patients. However, although these new trends bring exciting possibilities, they also come with a lot of challenges. The incredible diversity and complexity of these microbial communities makes it really difficult to accurately characterize their structure and their function. The vast number of microbial species, their complex interaction, their dynamic nature, all pose significant hurdles for data analysis and interpretation. To this end, maybe the biggest challenge in microbiome research today is the standardization of experimental protocols and analysis pipelines in order to ensure the reproducibility and comparability across different studies. Besides that, integrating and interpreting multi-omics data presents another significant challenge. The translation of complex microbial profiles into clinically relevant insights requires robust computational tools and bioinformatic pipelines, the development of which is vital for advancing microbiome research and harnessing the full potential of these approaches. However, challenges are not over. Another daunting challenge is distinguishing causality from correlation, especially in observational studies. The establishment of mechanistic links between changes in the structure of microbial communities and disease states demands sophisticated experimental design, including longitudinal studies and controlled interventions. Overcoming all these challenges is essential for elucidating the mechanisms that drive microbiome disease associations and inform targeted therapeutic strategies. To sum up, the evolving landscape of food microbiome research is marked by exciting trends, like the integration of multi-omics approaches, the emergence of microbiome engineering, and the development of personalized treatment strategies. However, these advancements come with challenges, including the complexity of microbial communities, the need for standardization in experimental protocols, and of course, the imperative to distinguish causality from correlation. Overcoming these challenges will be vital for realizing the full potential of food microbiome research in improving food safety, quality, and nutrition. Thank you.